Good afternoon, ma'am.
Good afternoon,
Ayan.
ma'am.
Good afternoon,
Okay.
ma'am.
Pag nag-response kasi kayo, I will know na naririnig nyo ko or okay yung aking audio. So thank you for responding. The type of the exam, kasi I am coordinating with the other F154 faculty handling the lecture component. So we discussed your concerns about the use of autoproctor. Ayaw nyo ng autoproctor? Sino yung mga estudyante ko sa lab? Most of you here are also my students in the lab. We tried using an autoproctor without a proctor, or hindi siya proctored na type, right? Mas madali siyang gamitin, di ba? We are looking at using Google Forms or yung quiz via LMS. So yun yung gagamitin. I want you also to understand that Fisheries 154 is a course na hindi siya pang-essay na type ng exam, kung mag-exam para sa assessment. No? This is an undergrad course, maganda yung multiple choice, yung mga ganun na type of exams, true or false and identification, kasi there are concepts that you really need to remember sa course na to. So, all right. So, 'di ba I gave an announcement that you will no longer submit outputs for module 6 and 7, no? And yung sa module 8, ano yung Okay, so yung module 8 natin wala na ring output kasi i-discuss natin lahat yung answers. Yung sa 6, 7, and 8 na mga output, i-discuss na lang natin via Zoom. So, hindi nyo na kailangan gawa ng output yon. I guess, yung lahat ng output sa sinubmit nyo from Module 1, 1, 2, 3, 4, 5, and Module 9 would be enough naman for me to assess your learning dito sa course na to, plus your exam. Do you agree? Okay na yon? Or gusto niyo pa yung mag-output sa 6, 7, and 8? Okay lang naman if you would insist. Pero ito, kasi sa module 9, you will be preparing a poster of a breakthrough microorganism. If you have checked your study guides, kasi binigay naman lahat dun sa Google Drive natin, lahat nung study guide andun na. So sa module 9, you will be preparing a poster. Pero I am thinking na instead of a poster, para ma-include lahat ng modules, or at least ma-include yung module 6 to 10, ay to 9, you will be making a video instead of a poster. Video of either a breakthrough microorganism. Yung breakthrough microorganism, sila yung mga microorganisms na ginagamit natin, like yung sa fermentation, sa biotech, yung mga may mga useful microorganisms. Yun. And also, either lang to breakthrough microorganism or yung microorganism that or agents of foodborne illness. Yun yung mga E. coli, yung salmonella, yun yung i-discuss natin later. So, instead of a poster, I want you to make a video of only one microorganism. It will be a group output. Like
Group out.
17 kayo, so there will, I will divide you into 3. So, there will be 2 groups na 4 yung members. So
Same groupings, ma'am.
5 groups. Ay, oo, 5 groups. Yes? Yes, Megan?
Same groupings sa dati, ma'am.
Depende sa inyo kung sino yung gusto nyong group mates. You just have to inform me ahead kung sino yung members ng group nyo. But before you post yung posting ng ating video will be on June 1. Earliest yun, kung kaya nyo matapos. But you have to submit first your video to me before nyo i-post. Titingnan ko lang naman kung kung okay na for posting. Halimbawa, ay, okay, yes, Angel.
Ano po, kalabog? Like,
ay, ano lang siya, very short video, 2 to 3 minutes. Actually, the shorter your video is the better. Kasi kung halimbawa, sa Facebook siya, ano din naman, parang dragging and boring kapag masyadong mahaba. Maganda yung mga videos na short videos, pero compact yung information. Like for example, you want to make a video on E. coli O157H7 strain. So first, i-introduce mo yon, di ba? Introduce mo yung microorganism, ano siya, and then ano yung reservoir niya, paano yung mode of transmission niya, ano yung type, ano yung food na most likely yun yung hinaharbor ng microorganism na yun, ano yung symptoms, and then yun na yun. So maybe you can make a video around 1 minute to 1.5 for that one microorganism. So pwede tayong gumawa nung parang meron siyang outline kung paano yung pagkakasunod-sunod ng video for that. But you should not forget to cite your sources if ever you have cited mga references. 500, okay. Sige, tawag 500. Class, I am proposing. I am not demanding. Okay? I am proposing. Kasi alam ko naman na most of you are having troubles din naman, troubles sa internet connection. Ang daming lamok dito sa office. Dito ako sa office. Ayan. So, 500.
fight, ma'am, sa 500, ma'am.
Fight, okay. Good. I like the spirit. Kasi ganito yon, yung ibang sections kasi, I think ginawa nila itong module 9 na poster. So para maiba naman tayo, kasi anyway, binawasan natin yung mga output, mag-video tayo, and gamitin natin yung social media. Oo. Okay?
What if, ma'am, yung bonus points, ma'am,
Sa live?
yung sa views na lang, ma'am?
Yung share is 500.
Yes, ma'am.
Yung bonus natin is sa views.
Yes, ma'am.
Sa views. So for every 100 na view, 1 point. Sigurado.
Pwede man, ma'am, daw.
Ay, ganito, wag niyo i-announce na bonus yun sa finals. Kasi baka yung ibang section walang bonus sa finals. Lagot tayo. So, sige yun. Kasi as much as possible, we're trying to standardize everything, and kung ano yung ginagawa sa kabilang section, ganun din sa kabila dapat. Pero dito sa mga output, binabago ko siya kasi masyadong madami. Masyadong madami yung outputs. Ayan. So, okay. 500 na shares. And then, for every 100 na view, for every 100 na views, 1 point in the finals. So, maximum of 300. Ang ibig kong sabihin, pwedeng lumagpas ng 300 yung mag-view. Yung bibigyan ko lang ng plus points is maximum of 3 lang. Maximum of 3 points lang yung pwede kong ibigay na bonus. Okay? Clear ba tayo doon?
Yes,
Kasi
ma'am.
baka mamaya hanapan nyo ako. Ang dami-dami.
Yes, ma'am.
Okay. Good. So, sige. So, pwede na kayong mag-create ng group, and then you give me your the name of your group mates. So, there are 17 of you in the class. So I am expecting na meron tayong 5 groups, no? 3 groups composed of 3 members and 2 groups with 4 members. So 8 plus 9, 17. Ayun. Okay? Sige, let me share my screen. It's on microbiology of food and water sa 5. Naka-project na ba yung screen? Ay, naka-slideshow na ba?
Yes, ma'am.
Okay. Sige, we will just go through this module, no? Because I have a recorded lecture for this one naman. Okay. So, teka, i-move ko lang itong ating beautiful faces. Pa-black tayo dyan. So, why do we need to study food microbiology? So, we know that microorganisms are causative agents of diseases in humans, right? And they are also the agents of food spoilage. Okay? Medyo nagugulo yung utak ko kasi I am also teaching Fisheries 160 no, that deals with fish handling, and katatapos lang ng lesson namin on fish spoilage. So ngayon naman, we're talking about food spoilage. Okay, medyo general tayo ngayon. So microorganisms can also be a source of food, and they can be exploited for food production. Ano yung example ng microorganisms that can be used for food, for production of food? Anybody?
Lactobacillus, ma'am.
Okay, lactobacillus. Good, Megan. Ano pa? Sige, as we go on, you will see that we can exploit microorganisms like your yeast, molds, and other bacteria for production of food. Okay? Yung mga fermented products natin, yun. Yung, possible yung fermentation process because of some microorganism. Hindi ko ma-remove itong mga faces natin. Teka. Ayan, nagbablock kasi siya dun sa content ng slide. Okay. So, here are the foodborne pathogens, no? The most common foodborne pathogens. So, we have the bacteria. No, later in the next module, we will be talking about the causes or agents of foodborne illnesses. And this would include the E. coli, O157H7, Salmonella species, Vibrio, Clostridium, Listeria, Campylobacter, Staph, and many others. And the Institute of Fish Processing Technology actually has a recently developed training module, no, for the detection of these foodborne pathogens in fish. Okay. We also have viruses, no? Examples of this are listed here: the enteroviruses, the Norwalk virus, and the hepatitis virus. So, what do you know about, bakit siya tinawag na Norwalk virus? So, Norwalk virus is a norovirus, no? And noroviruses are a group of related viruses that cause acute gastrointestinal illness. And itong Norwalk virus na to, kaya siya tinawag na Norwalk because there was an outbreak in 1968, no, in an elementary school in Norwalk, Ohio. So kaya siya tinawag na, yun yung naging name niya. Norwalk is not from a name of a person, but it's from a name of a place, in particular name of a school, no? And It was first identified, yung strain na ito, in 1970s. So next, we also have the prions. So prions are ano to sila, proteins, no, that are capable of folding or capable of dual conformation. And though it is not a bacteria or microorganism kasi nga protein siya, it causes severe illness, and up to this time uncurable siya kasi nga it has a tendency of sort of infecting other normal proteins to follow its conformation until it creates an aggregates of proteins. So, pag nagkaroon ng aggregation ng mga prions or tawagin natin silang abnormal proteins, it will now manifest an illness. So, we have a mad cow disease, no? Common na mad cow disease. And yung counterpart nyo noon sa humans is maraming lamok dito sa office, the Creutzfeldt-Jakob disease. So, yon. If you wanna check further on what prions are, you can watch this video. So ito tingnan natin kung pwede nating ma-watch ito briefly. Sige. Wala ito dun sa original na file na binigay sa inyo. Nag-ano ako nito, nag-add ako ng file. So nag-stop screen sharing muna ako, class. Watch natin first part lang ng video. Ang tingnan natin. So at least makita nyo lang kung ano yung prion. Ay, teka. Parang 4 minutes lang naman itong video. Pero tingnan na natin yung first 2 minutes. Naka-share yung screen.
Hi everyone and welcome to Biology Professor. Today we're talking about prions. Prions are a very interesting type of infectious disease. And they are so interesting because prions are actually proteins. When we think of infectious disease, we typically think of microorganisms, things like bacteria or fungi, also viruses. These are the things that we think of as causing disease, not proteins. And so when prions were actually first discovered, there was a lot of disagreement that this was even possible. But multiple experiments did eventually show that prions are proteins that can fold in at least 2 ways, and critically, one conformation that is one of the folding ways. One conformation can catalyze other prion proteins to fold that way.
So this is a prion, no? Mag-isa lang siya, but then it can catalyze other
Now, if you're
proteins to
interested in
aggregate.
learning more about catalysis and more about proteins and how they can act as enzymes, then see my video titled Introduction to Enzymes. But basically, catalysis
So ito na yung
is
aggregates.
simply when a protein acts as an enzyme. So enzymes are types of proteins, and they perform some function. And so in this situation, they are catalyzing another protein's change in shape. So here we have some drawings. I have the normal proteins in orange and a prion protein in pink. Now it's important for you to remember that the amino acid sequences of all of these proteins are the same. It's simply that the prion protein has a different shape, a different conformation than the normally folded proteins. And what happens is once one of these prion proteins is present, it can catalyze a change in shape of a normal protein. And this results in protein aggregates, specifically aggregates of the prion protein. So basically one prion protein catalyzes a change in shape for the normal protein, And that results in lots of prion proteins, so lots of the ones that have this particular shape instead of the normal shape, and then they begin to aggregate. And why is this a bad thing? Why does it cause disease? Well, it turns out that this type of situation, which we refer to as self-propagation, that this self-propagation of prion proteins making more prion proteins and then aggregating, it actually causes several brain diseases in different mammals. Here are a few that affect humans. There's mad cow disease, which you've probably heard of. It's typically linked with contaminated beef products. There's also Kuru. Kuru is a prion disease that is found in certain remote tribes of Papua New Guinea who practice funerary cannibalism. That is, they actually eat the brains of other dead tribal members and can get prion protein from those brains. There's also Creutzfeldt-Jakob disease, which is sometimes a genetic disease and is other times transmitted from person to person by way of brain grafts. And critically, all of these prion protein diseases are currently incurable, and they're also currently fatal. So these are serious diseases when contracted. Luckily, they are still fairly rare.
Balik tayo dun sa ating presentation. So that's prions, okay? So remember again that prions are not microorganisms but merely proteins, no? Ang ganda ng video na yon kasi very brief lang siya, short but very informative, no? Imagine meron pa lang pwedeng makuha dun sa Ruru na yung tawag ng disease doon kasi there's a tribe in Papua New Guinea na kinakain nila yung brain nung other individual and then they get the prions from those brains. Hay nako. Buti na lang it's not practiced here in the Philippines. So I hope you will not forget prions. Okay, so again, the mad cow disease is for the cattle, and the Creutzfeldt-Jakob disease is the counterpart of the mad cow disease in humans. All right. So let's proceed sa protozoans. So these are the agents of foodborne or the agents of disease, no? From food, Giardia lamblia, the C. parvum intameba, and the Cyclospora cayetanensis. Ayan. So I have a note here that usually the protozoans causes intestinal diseases. So the ones that they usually colonize and reproduce in the small intestine of humans. And of course, we have the helminths, the tapeworms, the flukes, roundworms, and the hookworms. Okay. Okay, so these are the common foodborne bacteria. Okay, so ano yung common dyan sa inyo na alam nyo na kung ano yung disease na kinokos niya? Anybody? Where can you get salmonella? Nag-move ba yung slide ko, class? Nandyan ba tayo sa common foodborne bacteria? Teka. Ay, ito pala muna. Sige, before we proceed to that one, no? So we have foodborne illnesses, no? Commonly referred to as foodborne poisoning. So there are two types, yung foodborne illness na yan. It could be called a foodborne intoxication or foodborne infection. Wala pala akong video. So foodborne intoxication refers to an illness no, resulting from consumption of an exotoxin produced by organisms growing in food, while foodborne infection requires consumption of the living organism. So can you see the difference between the two? Okay, pag sinabing intoxication, you are taking in the toxins, while infection, it's the living organisms that you have taken in, no, together with the food na kinain mo. So when food is ingested, no, it is the para sa intoxication, it is a toxin that's responsible for the illness, not the organism. And usually, the symptoms does not appear immediately. So it will take some time pa before mag-manifest yung symptoms. So the most common symptom associated with foodborne illness is diarrhea. No? Each pathogenic microorganism has its set of characteristic symptoms, and we will know more about those symptoms later. The severity of the foodborne illness depends on the microorganism or the toxins na na-produce ng microorganism. And also, dependent din yon sa amount of food that you consume, no? Yun yung dose of food, and the health status of the person eating that food, no, na poison, tawagin natin siyang poisoned food kasi nga it will cause food poisoning. Okay? And for individuals who have immunocompromised health condition, especially mga aged, children, pregnant women, yung may mga allergies, ganon, any foodborne illness may be life-threatening. Ayun. So, yung mga may mga underlying na mga illness should be very careful, no, in consuming food. They really have to check that what they're eating is free from those microorganisms. Okay. So, what are the common foodborne bacteria that you know? So later we will be discussing about Salmonella. Saan dito yung mga Bacillus? We will also talk about this. Campylobacter, Clostridium. I don't think Bacillus is included. Hindi siya included. E. coli, of course. Vibrio, Staph, Shigella, I think is included, and Salmonella. So yan yung mga pag-uusapan natin sa next na module. Okay? So what are the common genera of molds in food naman? So alam nyo ba kung ano yung molds na nagkukos ng spoilage ng ating bread? Okay, so rhizopus causes spoilage of our bread. Yan. Siya yung pinaka number 1 na culprit sa ating bread spoilage. But hindi lang limited yung action niya doon sa bread. It can also cause spoilage in strawberries and other berries, fruits, and vegetables. Okay, what else? Yung aspergillus. No, it's capable of producing mycotoxins, no? And these toxins are known for their strong and varied biological activities. So, magka-cause din ito ng allergies sa mga sensitive individuals, and these mycotoxins are also known to carry the most potent carcinogenic carcinogens. Ayun. Sige, tingnan natin. Next. Okay. So, Spoiled food is generally not harmful, but considered unsafe. No? Pwede na hindi siya tawag dito harmful, no? Kasi pwede na ang microorganism na present doon has cannot cause yung mga detrimental na illness. But for us na consumers, we already consider it unsafe. No? And food spoilage encompasses any undesirable change in food, specifically dun sa mga sensory attributes ng products or ng food. And food spoilage results from growth of microorganisms in food which alters food visibly and in other ways. Yung mga obvious na manifestations ng food spoilage natin, especially if it has been acted on by microorganisms are, first, may visible na growth. Excuse me. There will be change in color. There will be gas formation or magmo-moisten yung food, no? Yun yung mga obvious na manifestations of food spoilage. And it also involves predictable succession of microorganisms. So different foods undergo different types of spoilage processes, no? And toxins are sometimes produced. And so there are a lot of factors that may cause the unpredictable or predictable succession of microorganisms. Na halimbawa, ito yung component niya na microorganism, but once the food undergoes spoilage, its pH will change, no? Eventually, yung microbial flora niya will also change kasi malilimit na yung growth ng mga microorganisms that cannot tolerate the new pH of the food. So later we will talk about the intrinsic and extrinsic properties of the environment or food, no, and its effect dun sa microorganism. Naglalag-lag yung aking. Okay, so there is a wide range of bacteria important in food spoilage. So later, mga 9 ata yung pag-uusapan natin, no? And we have here, Pseudomonas can metabolize a wide variety of compounds. Bakit kaya sinabi na it can metabolize a wide variety of compounds? Sadali, class ha, inom lang ako ng tubig. So, Pseudomonas plays an important role in milk spoilage, no? And because they can produce many thermotolerant lipolytic and proteolytic enzymes. So, kaya siya sinasabi na it can metabolize a wide variety of compounds because of its thermotolerant lipolytic and proteolytic enzymes. Yung lipolytic can digest lipids, while the proteolytic can digest proteins. Yun. So kung yung product mo has high lipid and high protein content, yun, gustong-gusto yun ng Pseudomonas. Okay? It can also spoil a wide variety of food, no, including meat. Okay. So ito naman, ang third na statement is psychrophilic organisms can multiply in a refrigerator. So, di ba, what do we know about psychrophiles? Di ba, they are the, ano yung psychrophiles? Cold-loving, heat-loving?
Cold-loving, ma'am.
Cold-loving, okay. So, sila yung mga nagka-thrive at low temperature. No? So, to survive at temperatures close to freezing or freezing point of water, psychrophiles can evolve, no, or can do some important adaptations para makasurvive sila at refrigerated conditions. So one of those or some of the mechanisms that they can do are first to maintain, they should maintain their membrane fluidity even at low temperature. They synthesize cold acclimatation proteins. So, di ba yung isda merong anti-freeze proteins, ganon. So, sila, they are also capable of synthesizing cold acclimation proteins, excuse. They also do freeze tolerance strategies, no? And they also produce cold active enzymes. That's why they can still multiply in refrigerator or refrigerated condition. So yun yung mga psychophiles natin. And then next, most common genera include yan yung Erwinia, Acetobacter, and yun. Next is some of the most common spoilage fungi include Rhizopus, Alternaria, Penicillium, Aspergillus. Yun yung sinabi natin kanina, ang Rhizopus is a bread, no? And the fungi grow readily in low acidic moisture environment. So fungi prefer acidic than basic environment. Yung alam natin, maraming microorganisms prefer the basic, right? But fungi are tolerant sa acidic condition. Actually, they prefer acidic environment. And in fact, penicillium there's a species of penicillium that can thrive even at a pH of 1.0. Ayan. So take note that though many microorganisms prefer alkaline conditions, no, fungi can hardly survive at alkaline pH or alkaline environment. And most of them cannot even grow at pH 8.5. So yun. Pareho ba tayo ng tinitingnan na slides? Nakalagay sa slide is conditions for spoilage. Hello?
Yes, ma'am.
Okay, good. Okay. Kasi by the way, nag-record ako ng video na to. When I check my video, okay yung pag move ng slides ko when I was doing the video. Nung tiningnan ko na yung video, hindi pala siya umaalis yung doon sa recorded na file. So that's why medyo napapraning ako at nasasayang yung effort sa pag-record. Okay, so we know that in the earlier modules that we've tackled, microorganisms require certain conditions for optimum growth. So they require certain amount of moisture, pH, and oxygen, and amount of oxygen and temperature. However, for food naman, we have to also consider the water, moisture content, or water activity, and then the pH of the food, the physical structure of the food, oxygen, and temperature. Bakit importante yung physical structure? You have to know that, for example, in the case of meat, sorry, which one would spoil faster? Ito bang ground meat or the sliced meat? Which one will spoil faster? Si ground meat or yung sliced meat? What do you think?
Yung ground meat, ma'am.
Okay. Sino nasabat? Si Maureen?
Yes, ma'am.
Why do you think nga mauna si ground beef?
Hunch lang, ma'am. But I guess because mas exposed, mas dako ang area, surface areas, ng ground beef nga exposed sa air.
That's the answer. Okay, very good. Medyo choppy ka sa ako na. But thank you for your answer, no? So, ang ground beef provides a wider surface area for microorganisms to grow. Plus, since ground na siya, kumbaga, it's the nutrients, that's present in the meat can readily be assimilated by the microorganism. Kasi nga, soft na siya, ng ground na yung structure niya. So, it's easier for them to attack. Okay, do you agree? It is also important to look at the moisture or water activity. No? So by the way, ano yung pinagkaiba ng moisture content sa water activity? Sorry. Okay, yung moisture content defines only the amount of water in your food, no? Sorry Sorry class na-allergy kasi ako ngayon. So parang ubo-ubo ako. Pero wala akong COVID. Ano lang talaga? Allergy. So as you go on, no, as you go to higher fisheries courses, lalo na sa fish processing, you will encounter moisture content and water activity more and more often pag nasa higher years na kayo. And you will really have to understand the difference of moisture content and the water activity. So ano yung mas importanteng tingnan when you're with respect to growth of microorganisms? Moisture content ba or water activity? Guess lang, guess.
Moisture, ma'am.
Sana meron akong Mentimeter, no? Para papipiliin ko kayo, moisture or water activity. Sino nagsabing moisture aside from Angel? Okay, the answer actually is water activity. No, because the moisture content simply defines the amount of water in your food and ingredients. Okay, kung halimbawa you have your water and then kung gusto mo malaman yung moisture content ng food mo, you just dry your food, no, to a certain level, and then you measure the difference in weight. Yung nawalang weight, yun yung water. Now that's the amount of water in your food. But the water activity, no, refers to the bound water and the water that that would be able to predict ng stability ng food, and yun yung ginagamit ng microorganisms for growth. Okay? So it will explain, yung water activity level mo will explain how the water in your food will react with the microorganism. Teka lang. Excuse mo na ako Okay, I'm sorry. Nauubo talaga ako, guys. So, diba, most of the time when we talk about growth of microorganisms, requirement for growth, we're not dealing with moisture content, but we talk more about the water activity. So, dun sa inyong module 2 or 3, I think, sa requirements for growth, andun yung water activity na meron tayong pinakita doon na at this level, yung 100% kasi na water activity is 1, tapos pababa siya, 0.9, 0.8, ganon. So, yun yung makikita mo doon sa scale kung anong water activity yung dried fish, for example. Anong water activity dapat meron siya so that in order to limit the growth of molds, ganon, of bacteria and other microorganisms. Meron tayong ganon. I think in order to prevent the growth of fungi and molds, your product should have a water activity of 0.7 or at most 0.75. Ganon. Okay. Can you think of, if you're going to ask me, is it possible for a food to have a high moisture content but low water activity? Or AW, yung water activity natin, yan yung A, tapos may subscript na W. So, is it possible for a food to have a high moisture content but low water activity? Pwede ba yon na it has high moisture content tapos low yung water activity niya? Hindi ko naman nare-record yung sagot. Sumagot kayo, guys. Mag-guess lang, guess. Possible?
Yes, ma'am.
Okay.
I think so. Oo.
So, meron ba kayong ma-cite na example? Mahirap, no? Sige. Ang example dyan is your honey. 'Di ba? If you're going to look at honey, it's liquid. 'Di ba? Agree? Liquid. But it has a very low water activity. High in moisture content siya, but it has a low water activity. That's why yung honey natin, shelf stable. Pwede lang siyang ilagay sa room temp and it don't get spoiled. 'Di ba? Yung honey nyo ba, nilalagay pa ninyo sa ref? 'Di ba yung tinitindang honey, nasa bottle lang siya, tapos nasa ano lang siya, room temp?
Yes, ma'am.
So yun, yung explanation doon. No? Alright, so the microbes that we can eat. So sa mushrooms, sa cheese, no, yan yung mga fungi. Meron tayong yeast paste, yung bacteria spirulina, ayan, and sa algae we have chlorella. Itong spirulina and chlorella, common ito sa ginagamit na fish feed, no? But there are also products that uses this microorganisms and we are eating them. Okay. So these are the microorganisms that we can exploit, no? So we have the bacteria. Ayan. Andiyan naman siya sa notes natin, no? The yeast and the molds. So ang modern yogurt production, it involves culturing milk with live bacteria. Alam naman natin yun, di ba? So the bacteria produce lactic acid which coagulates the milk proteins, no? And in making so this protein coagulates, no? Makes the yogurt thick and slightly sour in flavor. Kasi nga merong may bacteria doon that acts on the milk. And the bacterial culture required for producing yogurt are Streptococcus and Lactobacillus. Ayan. So, for the yeast naman sa bread, beer, and mga wine natin, the yeast are used for the production of these products. And yeast is also used in as a leavening of bread, no? And it uses sugars and oxygen in order for the dough to produce more yeast cells and carbon dioxide. Kaya ano yung nangyayari sa ating bread kapag merong yeast? It what? Rices. Oh, si Megan, ano siya? Nagbe-bake. Nagbe-bake ka, Megan?
Nag-alantaw lang, ma'am.
Nagalantaw ka lang. Ayan. Ano naman yung role ng molds natin during cheese production? So, yung molds produces desirable flavors, appearance, and texture dun sa finished na cheese natin. No? So, yun yung mga role niya. So, you just read these lines here, okay? Para hindi na natin kailangang isa-isahin. Excuse. As much as possible, ayokong basahin yung laman ng PowerPoint kasi meron na kayong niyan yung kumbaga yan makikita na ninyo. I'll just input something kung meron tayong ma-input. Okay? Class, sorry medyo hindi lang talaga ako comfortable. So pag ganun ako, ganun. Okay. So microorganisms or growth of microorganisms in foods are controlled by several factors. Actually, for this slide, no, medyo kulang yung factors na nalista dyan. We have the extrinsic factors. So, examples of extrinsic factors are the temperature, relative humidity, gases, contaminating microorganisms, and this actually refers to the conditions of the environment or the storage environment ng food. No, food yung tinitingnan natin, and then yung extrinsic is the conditions extrinsic to the food. Right? Yung intrinsic naman are the excuse me. Composition, physical, and biological state of the food. So this refers to the physical-chemical properties of the food itself. Okay. In addition to the extrinsic and intrinsic, meron pa tayong mga implicit. So ano naman yung implicit na factors? These are the properties and interaction of microorganisms. So yung itong mga factors na to and kung paano mag-interact yung microorganisms, yun yung implicit na factors. And we also have processing factors. Kasi depending on the processing that you will do dun sa food or sa product, it will surely affect the microorganism or the microbial community present in your food. So ganun yun. So any change in the intrinsic and the extrinsic factors would lead to a change in your microbial community or microbial assemblage present in your food. Okay. So, ano bang example natin na pwedeng ma-cite? It could be possible, no? Like, for example, if meron kang raw fish, no? Kapag raw lang siya, you'll have this particular group of microorganisms that will grow on that food. However, if you, for example, marinate that fish, okay, you marinate it, so at some sort, you change the pH of the fish tissue and muscle. Ayan. So eventually, those microorganisms that cannot tolerate the low pH or acidic condition of your product will now be eliminated, no, dun sa fish mo. And those microorganisms that can tolerate the acidity of the product will now harbor the product. And if you store your product in a chilled condition, only those microorganisms that can tolerate the cold environment will now harbor or stay in and grow in your product. So ganun yung mechanism nung microorganisms sa food. Okay? So there are a lot of factors that affect their growth sa ating food. Okay. Actually, those factors are just elaborated in the following slides. No, we have the composition of food, pH of your food, presence and availability of water or yung water activity mo and moisture content, oxidation, reduction potential, the physical structure, and of course, the presence of antimicrobial substances. So, for the composition and pH, no, carbohydrates degradation did not result in major odor change, no. Ay nako, nakakahiya itong aking condition. Ayan, while proteins and or fats result in a variety of foul odors. Yun, there will be putrefaction, a proteolysis, and anaerobic breakdown of proteins, no, that would yield foul-smelling amine compounds. So yung mga ammoniacal na odor are caused by proteolysis. pH, no, impacts the makeup of microbial community and therefore the types of chemical reactions that occur when microbes grow in food. And also we have here low pH allows yeast and molds to become dominant. As we have what we have discussed earlier, mas gusto ng yeast and molds yung slightly acidic na environment. However, higher pH or slightly basic environment would allow bacteria to dominate your product. Ayan. So ito na si water activity. So in general, lower water activity inhibits the growth of microorganism. Okay, so water activity can be lowered by drying, or addition of salt or sugar. Ang addition ng salt or sugar, I don't know if we have discussed it here in this course, baka sa kabilang klase ko na naman. But the addition of salt or sugar, like in the case of marinating, nakapag-marinate na ba kayo, 'di ba? Ang ingredients ng marinating solution natin are salt, sugar, mga ganun, no? Mga paminta, ganun. So, by adding salt or sugar in your solution, 'di ba mas mataas ngayon yung if you will look at the osmosis na principle of osmosis, mas mataas ngayon yung magiging concentration ng solutes dun sa marinating solution versus your fish or the dun sa fish cells or tissues. So tendency is water inside the fish will leach out dun sa marinating solution. So in that way, bumababa yung water activity ng product or ng fish, no? Another is even under these conditions, spoilage can still occur by certain kinds of microorganisms. So itong mga osmophilic microorganisms, these are the ones that can thrive in a in a solution with high salt or sugar. And mga xerophilic microorganisms, they are the ones that prefer low water activity environment or food. Ayan. So we have the osmophilic and we have the xerophilic. Okay. Ayan, ito yung structure na sinasabi ko kanina, no? Yung grinding and mixing, ayan. So this increases the surface area, alters the cellular structure, and distributes microorganisms throughout the food. Kasi nga na-grind siya. And another thing, grinding would also allow yung mga microorganisms or mga enzymes, proteolytic enzymes that were initially packed in the ang tawag dito, yung mga organelles, no? Nakapack sila dun sa mga organelles nila. Will be released kasi pag halimbawa natamaan doon during grinding. So there will be proteolytic enzymes that will now act dun sa meat and it will facilitate growth and multiplication of your microorganism kasi mas may available food na for them to use for growth. Ayan. Okay, next, vegetables and fruits have outer skins, no, that protect against spoilage. However, spoilage microorganisms have enzymes that can weaken and penetrate such protective coverings. Iba talaga itong mga microorganisms. Micro but mighty. Ayan. So, even fruits and vegetables can be attacked by the microorganisms. So, pero yun sana yung protective structure ng fruits and vegetables. Okay? So, we have, anyway, mga antimicrobial substances, no, that's present in foods. And that include sa fruits and vegetables, we have the coumarins. It is a colorless, crystalline, solid antimicrobial substance with a sweet odor. Sweet odor na it resembles a scent of vanilla, but it has a bitter taste. So it can act as a parang chemical defense ng plants against predators. Ito namang lysozymes, no, present in cow's milk and eggs. It is actually present, it is an antibacterial enzyme found in many cells, tissues, and secretions from large range of organisms. Ayan. Paano siya nagiging antibacterial? It hydrolyzes the peptidoglycan of bacteria. Ayan. We also have the aldehydes and the phenolic compounds present in the herbs and spices. And we have the allicin can be found in the garlic. Ayan. So this also protects garlic from herbivores and parasites. Ayan. Yung isang clove daw ng garlic has approximately 10 grams, no? And can produce approximately 5 grams of allicin. So, imagine, kung 10 grams, parang 5%. 5% yung kanyang antibacterial na component, no? And this shows an enormous antibiotic potential, no? Contained in garlic bark. So 10 grams, 5 milligrams. Tama, 5%. Mataas na yon na antimicrobial na substance or percentage. Ayan, so let's now go to the extrinsic factors. Ayan, so the temperature. Lower temperatures retard microbial growth. Relative humidity, higher levels promote microbial growth. Lower RH would inhibit microbial growth. And then we know that oxygen promotes growth, no? However, aside from it promotes bacterial or microorganisms growth, it also facilitates I'm sorry. It also facilitates oxidation, yung oxygen. So if highly fatty yung product mo, be careful kasi it's possible na maging rancid yung odor because of oxidation. Excuse ulit ah. Okay. In order to manage yung environment of the product, no, and to eliminate or to control the condition or the mixture of gases inside the pack, meron tayong tinatawag na modified atmospheric packaging, yung MAP, no? It is a packaging system wherein it uses a wrap, no? Sinishrink yung wrap and vacuum technologies to package food at controlled conditions. So, vary yung amount ng carbon dioxide, ng oxygen, and other gases na pwedeng ilagay dun sa pack. So, at certain level ng oxygen, carbon dioxide, and kung ano pang gas yung ilalagay doon, it has a antimicrobial effect, and at the same time, it would limit oxidation of highly oxidative products or mga very fatty na products that easily gets oxidized. So, teka, yung aking battery is nag-signal na low bat. Okay, these are the sources of microorganisms found in food. So we have the soil and water, plants and plant products. Now, in the case of fishery products, no, yung soil and water na as sources of microorganisms pwedeng source ng ating fish, na i-relate natin sa fish processing. Ayan, yung plants and plant products, if we are using mga ingredients, no, na plants like yung mga pepper natin, yung garlic, onions, ayan. So, yun yung mga sources of microorganisms natin. Food utensils, na yung tools that we're using in processing, intestinal tract of man and animals. So, ito naman yung pwedeng matransfer yung microorganisms from the intestinal tract of man and animals if the workers or the person preparing the food does not follow good manufacturing practices wherein he or she should wash his hands before and after going to the bathroom, yung mga ganun ba? So, touching anything dirty, no, after touching anything dirty, you should always clean or wash your hands. No, mga food handlers, animal feeds, animal hides, air and dust. And yun yung mga sources of microorganisms. Ayan, let's go to fish processing technologies. Ayan. So first, I will talk about the microflora of dried and salted fish. No, so The preservative effect of drying and addition of salt is due to the withdrawal of water from the microorganisms. No? So when you dry or salt your fish, no, obviously, nare-remove yung water in the fish. So you are lowering the water content. Okay, so microorganisms and enzymes, inherent or microbial in origin, need water in order to be active. And at the same time, aside from removing the moisture, you are also reducing the water activity. So by reducing water activity, you are increasing the concentration of solutes, no, in the aqueous phase of the food. So tendency is yung pag mataas yung concentration ng solutes doon sa fish, for example, versus doon sa microorganism, yung water inside the microorganism will leach out doon sa cell niya and will go to the gradient where there is higher concentration of solutes. So dun sa food or sa fish mo. So yung microorganism mo will shrink. Eventually, it will shrink and die. Nakuha niyo yung point? No? So we have the fish na salted, for example, and then you have your microorganism present dun sa fish mo. So since salted siya, mataas yung concentration ng solute dun sa fish, ang tendency is yung moisture dun sa microorganism mo will leach out and will go to the environment na mataas yung solute concentration. Ang mangyayari dun sa bacterial cell mo it will shrink. Yun, mag-shrink siya. And eventually, it will die. Okay, most bacteria and molds can be checked by the addition. Teka, may natabunan. Yan. Can be checked by the addition of 5% salt. But halophiles can still grow. So, yung concern natin sa mga dried and salted products, yung mga problems dyan is yung ating mga halophiles. Kasi they don't easily get eliminated even at high salt concentration. Ano nga ulit yung microorganism na can survive even at very low AW or water activity? If we have halophiles, ano naman yung mga organisms that can tolerate low water activity food or products? Kalimutan nyo. Xerophiles. Xero. Diba? So there are two most important conditions in dried salted fish. No, meron tayong tinatawag na pinking. If you would observe dun sa mga dried products natin, minsan may mga nag-grow na mga pink na dati iniisip ko molds lang yon, pink molds, but actually ano yun sila, mga bacteria that causes pinking dun sa product. So madali siya i-remember kasi pinking yon. So this is caused by a group of halophilic bacteria, the Halobacterium and Sarcina. So they are killed by the addition of preservatives like sulfur dioxide. Ayan. So ano naman yung action ng sulfur dioxide dun sa halophilic bacteria? So sulfur dioxide is the one used in food and drinks industries for its preservative effect and antioxidant. So it can eliminate the Halobacterium and Sarcina sa iyong dried salted fish. Okay. We also have another one called DON, no? This refers to the brown discoloration of the fish or dried salted fish caused by the growth of molds, sporendonema. Ayan. So these molds have a temperature range of 10 to 37 degrees Celsius. Anong na-notice nyo sa optimum temperature ng molds na ito? Optimum growth niya is yung storage condition din ng ating dried fish, which is room temperature, di ba? So most likely, talagang mag-grow siya doon. And Sporodonema is not so biochemically active against fish flesh. So, hindi niya masyadong kinukonsume. Hindi niya sinisira yung flesh. Unlike the pinking bacteria na talagang kakainin nila yung flesh ng fish. So, dito sa brown discoloration, magdi-discolor lang talaga yung meat, pero hindi mawawala yung meat. And it will not cause destruction in the structure of the product. Discoloration lang talaga siya. Okay? Okay, next is low temperature, icing, chilling, and freezing. So this one, the principle in this is it will freezing or chilling and icing retards or stop the growth of microorganism in food, and different temperature requirement for growth of its, teka lang, natabunan na naman. Different temperature requirement for growth of each group of microorganisms, and this prevent the growth of these microorganisms, but the metabolic activity still continues. So that's why it's not a guarantee na kapag frozen yung product natin, it will no longer get spoiled. Mag-spoil pa rin yun. Kasi aside, although medyo na-suppress yung growth or na-prevent yung growth ng microorganisms, yung enzymatic activity still continues. Ayan. So icing and chilling, the temperature is just above freezing, so 0 to 5 degrees Celsius. No? It offers or provides temporary preservative effect to the product, especially in fish. And the principle of this is cooling of the environment would prevent microbial and enzymatic activities. Hence, it also prevents spoilage, but not indefinitely. Anong sinasabing not indefinitely? Kasi hindi niya kayang tuloy-tuloy, no? It can only suppress, but it cannot totally stop. No, slowing down the enzymatic processes and microbial growth. Okay. Ayoko nang medyo madaming animation na PowerPoint. Ayan. So in the case of freezing naman, it's the definition of this, the temperature is just below chilling. So the temperature is around -18 to as low as -30. So for your information, yung freezer natin sa ating household ref is around -18 to -20, no? And if you need to store your products in much lower temperature, you need to use an ultra low freezer. So merong mga ultra low freezer na meron siyang temperature na as low as -80. Ayan, yan yung mga ginagamit sa cell lines, sa cryopreservation ng microorganisms. Ayan, and if you wanna store your mga products for very long time, pwede mong i-store doon. However, though mababa yung tendency na mag-grow yung microorganism, very low temperature storage can also cause damage dun sa sensory attributes ng products. And you will know more about that later on. Okay? So, freezing could prevent the growth of microorganisms. They kill bacteria due to osmotic damage and intercellular growth of ice crystals. It can also increase the concentration of dissolved substances in the unfrozen state. Hence, it decreases AW. And paradoxically, freezing is a well-recognized method of preservation. Now, paano siya nasabi na it increases the concentration of dissolved substances? No? Kasi, yung water na initially present dun sa fish or sa product mo will now become unavailable for growth ng microorganism kasi nga nag-frozen. So that's why, in a sense, you have decreased the water activity of the product. Kasi nga, nag-frozen yung kanyang water that's supposed to be used by the microorganism for growth. Okay. So please note that freezing of cells depletes the water that's usable for growth. Yung sinasabi ko kanina that it will cause in a decrease of water activity, no? And also, I have a note here that freezing induces temperature shock in some of the microorganisms. This is true, no? Especially for thermophiles and mesophiles. And more cells die when the temperature decline above freezing is sudden than when it is slow. So kung abrupt yung pag cool nung product, just in the case of when you do blast freezing, kasi 'di ba when you do blast freezing, chilled lang yung product and then immediately ilalagay mo siya to negative 30 degrees Celsius in just 2 hours. So, pag ganun yung rapid freezing yung ginawa mo, it will cause parang thermal shock doon sa temperature shock doon sa microorganism. Even the mesophiles and the thermophiles. Okay? It can also cause metabolic injury to some microbial cells such as Pseudomonas. No? Some bacteria have increased nutritional requirements upon thawing from the frozen state, and as much as 40% of a culture may be affected. And here, freezing. Ayan. So the cells that are still viable immediately after freezing can or freezing die gradually when stored in the frozen state. Hindi siya nagmo-move. Ayan. So naintindihan niyo yung previous yung last line, no? The cells that are still viable immediately after freezing. So when you do blast freezing, meron pang cells na buhay, no? It will eventually die then when you store the product in a frozen state. Kapag maintain mo siya doon sa negative 30 or you store it to a very low condition like negative 80, mamamatay na rin eventually yung initially viable na cells. Okay. Okay, so there are factors that affects the lethality of freezing, no? And especially if yung microorganism mo is in the logarithmic phase. Ayan. However, it has, ayan. Okay, note here that if you're going to rank, ito kasi ni-rank na yung Salmonella, SRUs, and vegetative cells of Clostridia, no? In terms of survival, no, sa sa freezing. So, do you agree na yung vegetative cells ng Clostridia ay mas matagal mas matagal siya mamatay at frozen condition?
Yes, ma'am.
Okay. Sige, i-share ko itong notes ha. I don't know if makita nyo itong notes na to sa inyong PowerPoint na na-share sa inyo. Naputol ata ito. So, we have here, Salmonella is a gram-negative rod, no? And so, yung sinabi kasi dito, cocci are more resistant than rods. Kasi, The higher the surface area of the cell, the higher the membrane damage owing to extracellular ice crystal formation during freezing. So yung rods kasi, 'di ba, mas malapad sila. So mas malaki yung surface area nila na na-expose dun sa low temperature, kaya mas madali silang mamatay, no, as compared with the cocci. So theoretically, bacteria can grow at all temperatures between the freezing point of water and the temperature at which protein or protoplasm coagulates. So somewhere between the maximum and the minimum points lies the optimum temperature at which the bacteria grow best. But syempre, at freezing condition, hindi yon yung optimum na temperature for growth ng most microorganisms, okay? So the bacteria differ in their capacity to survive during freezing. So with the cocci being generally more resistant than gram-negative rods. So tama yung sequence na nakikita natin. Okay? For the food poisoning bacteria, Salmonella, they are less resistant than Staphylococcus aureus or the vegetative cells, whereas endospores and food poisoning toxins are apparently unaffected by low temperature, even freezing. Obviously, no, kasi yun naman yung purpose, no, na may mga spores sila that they produce into spores. Okay, so strictly speaking, kahit sinasabi natin kanina na freezing is one of the, ano yung sabi kanina? Freezing is one, teka. Saan na yun? Freezing is a well-known, is a well-recognized method of preservation. Strictly speaking, freezing should not be regarded as a means, no, of destroying foodborne microorganisms kasi hindi siya ganun ka-effective. Okay, later you will know ano yung pinaka-common na means on how to control the growth of foodborne pathogens. Anong oras na ba? Parang ang bilis ng oras natin. Diyos ko. Hanggang anong oras tayo, class? 3 o'clock lang ba? Sige, nasa slide 30 na tayo. So factors affecting lethality of freezing. So we have the time of storage in the frozen condition. So those that are still viable gradually die on storage. Tandaan nyo yan. And also the nature or the kind of food. No? Nature and composition. So ang glycerol, amount of glycerol, egg white, and meat extracts increase the viability of microorganisms. In fact, if you want to keep your microorganism longer, yung pure na microorganism ilalagay mo sa tube, you need to add glycerol para ma-cryopreserve mo siya. You can keep it for a very long time at negative 80 degrees Celsius. Glycerol will prevent the destruction of bacterial cells during low temperature storage. So basta merong glycerol, egg whites, and meat extracts, ini-increase nun ang viability ng microorganism. Okay? High water activity and low pH, this decreases the viability of the microorganism. Ayan. Okay, so mga high temperature techniques, preservation techniques, we have the blanching. Ano yung blanching? Yung sinasawsaw mo lang? Yung method ng pagluluto nung talaba, 'di ba? There's the boiling water and then lalagay mo sa screen yung talaba, iba-blanch mo siya. So that
Madalian lang, ma'am.
Hello? Yes?
Yung madalian lang, ma'am.
Yes.
Ano.
Correct. That's correct. Madalian lang siya. Sinawsaw mo lang tapos nilift mo. So that's blanching. Yung effect nun is you are denaturing the enzymes. Next is pasteurization. So this destroys the pathogens, no? Ang pasteurization natin is around 70 to 80 degrees Celsius. And then we have the commercial sterilization. Pathogenic and toxin-forming organisms are destroyed during canning, bottling, and aseptic processing. Next, we have the sterilization or complete destruction of all the microorganisms, including both the vegetative cells and spores. So when we do canning, we only aim for commercial sterility of the product. We don't do total sterilization. Kasi this, yung purpose lang naman natin dito sa commercial sterilization is to kill the pathogenic and toxin-forming organisms. We don't need to achieve the total sterility kasi it would, mas matagal na pagsa-sterilize yung kailangan mo, and it would entail more energy. It will be more costly. So if you are doing canning for commercial purposes, you only go for commercial sterility. Kasi you can already achieve the safety level or the safety requirement of your product with a minimum cost kung commercial sterility lang yung i-achieve mo. Whereas if you go into to sterilization talaga, it would incur additional cost on the part of the manufacturer. So, ganun yun. Later, when you take Fisheries 156, commercial sterilization and absolute sterility will be discussed. Ayan. Absolute sterilization. Ayan. So, we have a flat sour spoilage here. No? This is common in your canned products. Now, the ends of the can remain flat, and spoilage cannot be detected unless na-open mo yung product and natikman na, oh, it's sour. Ayan. So, there is production of carbon dioxide and water. No? Teka. Di siya nag-move. Ayan. So these are the microorganisms capable for the flat sour spoilage. And we also have the TA spoilage or the thermophilic acid spoilage. There is a production of carbon dioxide and water, and it's caused by Clostridium, no? Thermosaccharolyticum. Ayan. Ang haba. Pwede itong pang spelling bee. Ayan. And we also have sulfide spoilage, no? And the most obvious manifestation of this is the production of hydrogen sulfide from putrefying proteins. Ayan. Parang ang bilis ng 2 hours. So smoking has bactericidal effect. Okay? And however, non-spore forming rods can survive, no, and which can be introduced during handling. So, ito yon, they can grow at refrigerated temperatures. Fermentation. Ayan. Ano yung process ng fermentation? You allow the growth of desirable microorganisms. No? And ano yung principle niya? The presence of the organic acids has antimicrobial effect. No? And the acidic nature of the product is already below the growth range of most pathogenic microorganisms. And this would allow metabolic inhibition by the undissociated acid molecules. Ayan. So, ito yung mga antimicrobial products of fermentation. Ayan. So, I'm sure there are some of you who have already submitted their concept map. Okay? So now we have ilang minutes? Barely 10 minutes. Sige. Nag-end ba ako nung I-share ko lang yung gusto ko kasi class answeran natin yung yung i-open ko muna. Yung sa module 6. 'Di ba hindi kayo nag-output dito? Meron ba sa inyo na nag-study na ng module 6? Sana naman meron. Class, remember ha, yung exam natin are all taken from the PowerPoint slides. So, andyan lang lahat yun. Plus, if some concepts not included in the PowerPoint slides are given emphasis during lecture, so yun. Basta hindi ako nag-include ng anything sa exam na hindi natin na-discuss. Para fair din naman sa inyo. Okay? And na-notice nyo doon sa first long exam natin, I added some supplemental lectures, no? Ano yung ma-remember nyo na lecture natin na dinagdag ko sa module natin?
Yung about sa immunization and vaccination.
Yes, yon. So, kasi importante naman talaga na you as fish microbiology students of taking fish microbiology should also know, no, kung paano pinoproduce yung vaccines and paano ba nagko-cause ng protection yung vaccines. So, at least alam niyo. Okay? So, sa second part ng ating klase, dito sa module 5 to 9, I don't know kung ano yung mga additional lectures natin. But I think we will stick with what's given to us. Kasi parang kinat-short nila yung SEM. I was thinking na meron pa tayong 4 meetings, pero meron na lang pala tayong 3, I think. 3. So, 31, 7, and 4. 31 and 7 na lang. Dalawa na lang pala. Ako matatapos na yung SEM. Gusto niyo na rin matapos yung SEM? Siyempre, no? Gusto na, no? Stressful. My God. Sige. So, you go over, teka, na-share na ba to? Okay, makikita nyo kahit hindi ko siya hindi ko siya i-project, i-slideshow. Okay lang. Can you see the...
May ang font, ma'am, hindi kita nang gawa.
Hindi siya kita on. Teka lang. Okay. Okay. Hindi ko na siya i-lecture lahat ng slides, no? Kasi baka ma-overtime tayo kasi meron naman kayong class. So, what's important to note here is ito. Foodborne diseases are classified into 2. No, we have the food intoxication and infection. That's what we have also mentioned earlier. So, intoxication siya if the exotoxin are the ones causing the symptoms or the illness. And infection if it's the microorganisms itself grows inside the body and causes the symptoms or illness. So, ayan. So, food intoxications can be classified or can be caused by biotoxicants, metabolic products or exotoxins, and the poisonous substances. So, ito yung mga poisonous substances, yung mga histamine, yung mga, yun yung tinatawag natin na scombroid poisoning. Tapos yung mga aflatoxin and yung mga toxins na nakukuha sa pag merong red tide, yung mga ganun ba? So, yun. I don't know if it's included in here, pero yun yung mga under sa poisonous substances. Okay? For foodborne intoxications, most of the symptoms include short incubation. It has short incubation period with lack of fever, absence of fever. No? Well, however, sa food infections naman, usually merong fever. Ayan. Ayan, meron siyang fever. Yan yung obvious na pagkakaiba nila. So, in order to help you study the module easily, you prepare a table like this, no? You list the causative agents, and then you note if it is a gram-positive or gram-negative, and then the shape, the conditions for growth, merong ganyan, the types of food, the toxins that they produce, and the symptoms. So if you have this table, magiging madali sa inyong tingnan no, yung characteristics ng microorganism. Hindi mo siya kailangang i-memorize. You don't need to memorize each microorganism, but at least you familiarize them. No? So these are the foodborne pathogens no, that causes intoxication. So we have the Staph aureus causing the staphylococcal poisoning. We have the E. coli O157H7. It's the one that causes hamburger disease. Now you have to check kung ano itong O157H7. Bakit siya nagkaroon ng pangalan na ganito, okay? So at least malaman niyo bakit siya na merong code na ganyan. Meron siyang ganyan after the E. coli name. Clostridium botulinum, this is common in the canned products. Now, it causes botulism. It is very fatal, no? So, yung Clostridium perfringens, they are the ones that causes canteen disease. So, basahin niyo isa-isa yung mga slides, no? Kasi it's around 88 slides, I think. So, hindi natin siya kayang kunin in one meeting. But since naka-cut short nga yung SEM, parang you need to do it on your own time, no? Alright. So, pwede ba nating diretsuhin na yung sa activity? Ano yung oras? Sige, feeling ko kaya natin siya in 5 minutes. Sige, ano ko muna ha? Skip ko ito. Puntahan natin dun sa, ano ba yan? Ayan. Dito tayo diretso sa activity. Okay. Can you see the slide na may case study number 1? Nakikita nyo?
Yes, ma'am.
Okay. So, sorry ha. So may class ba kayo next? Sandali lang to, mga 5 minutes. Para lang ma-present ko na agad. So, ito yung case study number 1 na supposedly is output ninyo. No? So, a physician suspects one of his patients has botulism, a potentially fatal illness. As the CDC or the Centers for Disease Control and Prevention epidemiologist assigned you to the case, it is your responsibility to determine the source of illness and to suggest means to prevent others from becoming ill. This involves interviewing the patient, determining potential exposure sources, determining what should be tested, interpreting laboratory results, helping to determine necessary actions to prevent others from becoming ill, and helping to determine actions necessary to prevent future occurrences. So, ito, you travel to the hospital to interview the patient, and you gathered to gather more information. So meron kang nakuhang table. Later, makikita mo, by the time you arrive, the patient's condition has worsened dramatically such that he is unable to communicate. He lives alone and works from home, and consequently, there are no close contacts available to provide information about his eating habits. Credit card records show no restaurant charges in the past 2 weeks. So ano yung implication kapag no credit card records? Ibig sabihin, hindi siya kumain outside. Okay? So dishes in the dishwasher are clean. Inspection of his home refrigerator, cupboards, trash, and recycle bin yields various items listed in the next table. And using the data provided, you, as the one assigned to this case, should complete the table by indicating whether or not the food found in this patient's home have the potential to produce Clostridium botulinum growth and toxin production. So provide explanation for your determinations. Ayan. So ayan, meron kang nakitang ground meat, no? Opened, yes. So suspected food ba siya? No, or why not? Why or why not? So what do you think? Yung ground meat, suspected food ba siya? Tingnan natin yung ating, teka ha. Kasi, ayan. So, yung ating ground meat, no, is yung answer natin suspected food, it's a yes. Because, naka-vacuum pack siya in a tray. So, ibig sabihin, anaerobic. If you would check the characteristics of Clostridium botulinum, no, it grows at anaerobic condition, and since nasa vacuum pack siya, ito yung pH niya 6.7, slightly acidic, so suspected siya. And then yung strawberries, no. Okay. And then yung pineapple chunks, no din. Kasi very acidic, hindi mag-grow doon yung microorganism na yon. And hindi mapoproduce yung yung toxin. Chili beans, tingnan nyo yung ano niya, canned siya, no? Canned, and yung acidity niya is slightly acidic. And then anaerobic, kasi canned nga. So, low acid. So, posibleng yun yung source. And then, tingnan natin yung ketchup. Very acidic. So, hindi yon. And then, look at the milk. No? It's a plastic jug. Hindi rin siya, no? Kasi hindi naman siya anaerobic. Yung orange juice, nasa carton. Hindi rin. Kasi very, very acidic. Yung grape jelly, hindi rin siya. Vegetable soup, ayan, canned. So, yung Clostridium kasi nagtathrive siya sa anaerobic, no, pag canned. And then yung temp pH niya is 6.3. So, most likely, isa ito sa mga suspected na food. Next is dried spaghetti. Hindi rin, no? Yung tomato sauce, very acidic, hindi rin. Soda, hindi rin. Okay? So, I hope nasundan ninyo yon. And then, eto, 28 kindergarten children. Ay, teka, hindi ata na-move yung slide ko. Bakit may kuris-kuris? Ayan. Okay. 28 kindergarten children had 7 adults visited a raw milk bottling plant where they were given ice cream and raw milk. 3 to 6 days later, 9 children and 3 adults developed gastroenteritis. So the food, only the foods eaten by all these children, ill and well, no, yung mga may sakit at walang symptoms, were in the school provided lunches. No one else in the school became sick. So stool cultures showed 1 bacterium in common to 9 of the ill children and not present in samples from 9 well children. So this bacterium is a curved gram-negative rod, and it neither ferments nor oxidizes glucose. So ibig sabihin, nag-conduct sila ng biochemical test. So ito yung mga questions. Was it food infection or food intoxication? So what do you think? So the answer here would be food infection. And what item was contaminated? Posible yung raw milk, no? And what could be the etiologic agent of this outbreak? So posible na yung Campylobacter. So ang problem natin ngayon, hindi natin na run through si Campylobacter, no? So what would be done to prevent this type of outbreak? Pag milk, dapat na-pasteurize si milk. Okay? And yun, ano yung source ng infection? So yun na, yung raw milk. Okay? Okay, so that's all for module 6 and 7. I will upload the recordings ng module 5 and 6 pala. Ayan. So, most likely class, we will be having sunod-sunod na synchronous classes until June 7. Saan na ba yung aking calendar? Yan. So next meeting is May 31. Next meeting after next is June 7. So we will be having synchronous meeting sessions until sa last day ng class natin. Sorry, kailangan natin 'yon. Hindi lang tayo magsisynchronous kapag may brownout. Okay lang. Kasi we need to make habol, no, para maubos natin yung course content. Ayan. Thank you. Stay safe. Bye-bye. See you on Monday next week.